Good morning, students. We have begun with a new unit, that is molecular basis of inheritance. This is the wonderful chapter you have in all of your syllabus. I am not saying others are not good, but this is very technical and very informative and useful in future, right? When some chapters I give you that they are very important, it's not just based on the questions and other things appearing. Apart from that, they are highly applicable subjects. Means your future you can make it with these chapters, right? As I told you, started with the genetics with Mendelian inheritance, right? Now we are going to continue with that. It's called the Mendelian, that's the molecular biology or molecular basis of inheritance. What is this all about? It is about how exactly inheritance happens, molecular level, not externally, molecular level, how there is inheritance, that is molecular biology. So first part in this is about the nucleic acids. So let's begin with the nucleic acids. So what are the nucleic acids? Nucleic acids are actually DNA and RNA. These were first discovered by scientist Frederick Mischer, right? This person discovered these nucleic acids in the pus cells, the nucleus of the pus cells he identified. There are two types, DNA and RNA. Now, major nucleic acid what we learn is only DNA. So I will be giving mostly about DNA. At the later stages I will give you something about RNA. So DNA is in long form. We call it as Deoxyribonucleic acid. So DNA is otherwise called Deoxyribonucleic acid. Okay, this is DNA. Now, what is the DNA? Where it is found? It is most of the higher organisms. This is the hereditary material. Means we get our characters from parents to offsprings. We get it from our parents through DNA. DNA is a hereditary material of every cell found in the nucleus of the every eukaryotic cell in their chromosome, right? So where is DNA found, right? Suppose you think that this is the cell, this is the nucleus, this is the chromatin. So take this and observe, you get the chromosomes like this, chromosomes, okay? These chromosomes internally will have the DNA, okay? This is the DNA, double stranded DNA. So now this DNA I am explaining you, this DNA, where it is present? This DNA is found in all the cells of human body or all higher organisms. In lower organisms also, almost every organism contains DNA. But how is it in lower organisms and in higher organisms? First thing, bacteria contain DNA, double-stranded DNA. But in bacteria, the DNA will be found in the center in the form of just a coiled circular structure. This is a circular double-stranded DNA in bacteria. It is in bacteria. Okay. In lower forms, this is called the nucleoid. They don't have the nuclear membrane, right? But if you get any cell, imagine either a plant cell or animal cell. If you take the plant cell, imagine the plant cell. Inside this, you get, apart from the main nucleus, you get chloroplast, right? The chloroplast will have membrane structure. Take the chloroplast and observe under microscope, you find its own DNA apart from its thylakoid membranes. The thylakoid membranes are there everywhere, I told you. Apart from this, you find one double-stranded DNA. This is DNA. It's a naked DNA. Even same thing happens in the mitochondria. In mitochondria, you got cristae, right? Cristae. Inside this, you will have the DNA. This is again a circular DNA. This is a primitive DNA. And in some of the animal viruses, you get double-stranded DNA. Some will have single-stranded DNA. But plant viruses normally contain RNA as a genetic material. Okay. Now we talk about the deoxyribonucleic acid of higher organisms. All the higher organisms contain the double-stranded helical structure of DNA. That I am going to explain you today about the structure of double-stranded DNA. Okay, let's begin with the DNA. Deoxyribonucleic acid. But along with that, I will give you the comparison also with RNA. Okay, DNA and RNA will compare and give you the differences. But before anything, let me give you what a nucleic acid consists of. So every nucleic acid has three components. One is a pentose sugar and a nitrogen base and a phosphate group. Okay, these are forming a unit called as nucleotide. So nucleic acid is the chain of nucleotides. It's called the polynucleotide chain. Means this all thing, the sugar plus nitrogen base plus phosphate forms one nucleotide. Like that you get many such nucleotides together. This is called as a polynucleotide chain. Sugar plus nitrogen base plus phosphate. Sugar plus nitrogen base plus phosphate. This is the polynucleotide chain forming the nucleic acids. Right? Now it is a time for you to understand what is the structure of nucleotide, how it is. Each nucleotide, if you understand, the remaining thing will be easily understanding. So one of the structure I will give you, chemical structure I will give you, how it looks like in RNA or DNA. So let me give you first RNA here, next I will write DNA here. Okay. First part what I am giving you is pentose sugar. How is pentose sugar? This is in RNA, it is called ribose sugar. Sugar is pentose, having 5 carbon. This is called deoxyribose sugar. Okay, how is ribose sugar? It has the pentose structure like this. Okay, OH, H, OH, H, OH, H, H, CH2OH. This is the ribose sugar, right? Ribose sugar, you can see, you can number them. First carbon, second carbon, third carbon, fourth carbon, fifth carbon. 5 carbon sugar, it is a pentose sugar called ribose sugar. What is the difference between this? Deoxy means there is no oxygen. From ribose sugar, you are going to remove one oxygen. Right? Again, I am writing the same thing here, but what is the difference? You have to make it out. Okay, here is a OH group, here is H group, here is H group, here is H group, here is OH group, here is H group, H and CH2OH. So, which carbon lack an oxygen is? Here see, second carbon has OH and second carbon has H. Therefore, this sugar's name is deoxyribose sugar. Sugar having less oxygen from ribose sugar is deoxyribose sugar. Okay? Then, second thing is about the nitrogen bases. Nitrogen bases are studied under 2 headings. First one are called as purines. Second one are called as pyrimidines. Purines present in RNA are adenine and adenine, written as A, and guanine, G. Okay, A and G. Even here in DNA you get adenine and guanine as purines. But pyrimidines found here are cytosine, C, and uracil, that is U. Here you get cytosine, C, but instead of uracil you get thymine, T, right? So, adenine, guanine, cytosine, thymine are the nitrogen bases of DNA. Adenine, guanine, cytosine, uracil are the nitrogen bases of RNA. Okay? Now, phosphate group is found in both of them. Then other differences I will give you. I will not write it. You just have to note them down. What are the other differences? DNA is always double-stranded, having 2 strands, right? This will have, whenever you write DNA, there will be one strand and the other strand. Okay, here it will be attached with the help of nitrogen base. This is DNA. RNA is normally single stranded. This is RNA. Okay. Now, another thing, there is a base pairing happening in DNA, right? So what happens? How exactly bases pair? Right? If I say DNA, here it will be having the strand with sugar phosphate backbone. Again, next strand, sugar phosphate backbone, connected by adenine to thymine, guanine to cytosine, and so on. ATGC pairing. So, one more characteristic I told you, in DNA you get A to T double bond, G to C triple bond. Base pairing happens. But here, normally it is single stranded, base pairing will not happen. But suppose this RNA turns like this and pairs here. That's called a false pairing. If it happens, that time how it will pair? It pair adenine with uracil and guanine with cytosine. That is the difference, right? Other differences. So let me list them all. RNA, ribose sugar it contains, and DNA contain deoxyribose sugar, right? Second is nitrogen bases. In RNA you have nitrogen bases are purines, adenine and guanine, pyrimidines, cytosine and uracil. In DNA you have adenine and guanine purines, cytosine and thymine as pyrimidines. And normally I told you DNA is double stranded structure, RNA is a single stranded structure. If the DNA is double stranded structure, then how it is pairing? Normally the sugar and phosphate from the strands, sugar and sugar are connected by nitrogen bases. Nitrogen base adenine pairs with always thymine, or thymine to adenine, vice versa is true. Adenine to thymine or thymine to adenine, right? AT, TA, both are same. Then guanine will pair with cytosine with 3 hydrogen bonds, or cytosine to guanine 3 hydrogen bonds. Adenine to thymine is 2 hydrogen bonds, guanine to cytosine is 3 hydrogen bonds. A double bond T, G triple bond C, or T double bond A, C triple bond G. That is ATGC is pakka pairing happening in the DNA. But what about the RNA? In RNA, you will not get pairing because it is single stranded, right? Take it, always it is single stranded. But if it pairs by false pairing, if the single strand will turn back and pair, that time adenine pairs with uracil, guanine pairs with cytosine. That is the major difference. That means nitrogen bases in DNA and RNA mainly differs in pyrimidines, where thymine of DNA is replaced by uracil in RNA, or uracil of RNA is replaced by thymine in DNA. What are the other differences? Another difference. So this will be your homework. Differentiate between DNA and RNA. Next difference is DNA is normally hereditary. It will transfer the characters from the parents to the offsprings. Therefore, it is called the hereditary material. But that's not true with RNA. RNA is normally non-hereditary. I told you, except for certain plant viruses. Plant viruses will have RNA as hereditary material. But mainly, majority of the organisms, they will have DNA as a hereditary material. Now, what next? DNA will be controlling the whole activities of the cell. Means everything, suppose one cell, it has to produce proteins, or it has to replicate, it has to double. Everything is controlled by DNA. We call that the master macromolecule of the cell. Master molecule of the cell. It will control every activity required for the cell. The same way, this DNA will have the ability of self-replication. Means one DNA can form 2 DNAs on its own. No other thing involved in a DNA will undergo self-doubling. That's an important characteristic feature of DNA. DNA will undergo self-replication or self-duplication. RNA will not do. But RNA, who is producing? DNA is producing RNA. DNA only will produce RNA. RNA is not self-produced. Right? Then other things, DNA is found in the nucleus in most of the organisms. In some it is found in the cytoplasm also. You would have seen in bacteria, some plasmids are there, extra chromosomal DNA will be there. That is extra chromosomal DNA, circular DNA found in the cytoplasm. Mostly the DNA is found in the nucleus, but sometimes it is found in the cytoplasm also. Sometimes it is found in certain organelles also. Which organelles I told you? One is mitochondria, one is chloroplast. Both contain the DNA as a naked DNA. They are not covered by anything. Right. Then RNA will be produced by the DNA, and it is not doing other activities than helping for protein synthesis in the cell. Right. But where is RNA located? It is produced by DNA, so it is found in the nucleus. It is released into the cytoplasm, so it is found in cytoplasm also. So DNA, majority of DNA is found in the nucleus. Very exceptional way you study, it is found in the cytoplasm. But coming to RNA, RNA is produced by the DNA, so it is in the nucleus. It is there in the cytoplasm because it works in the cytoplasm. What are the other functions of DNA? DNA takes part in or helps, or it will guide the process of protein synthesis. Right? When I talk about protein synthesis, I will explain the details of this. Means whenever any body or any cell require proteins, this DNA will produce one copy of itself. What protein has to be produced, it will send it, and RNA will help in producing this protein in the ribosomes. Right? So DNA is the major one which is guiding protein synthesis, but RNA are taking part in protein synthesis. So many lists of differences I gave you. You need to write the differences between RNA and DNA. That will be your homework. I want you to finish and send me. Okay? Done. Now we will move on to the structure of DNA. This is the most important thing. First you write the differences. Structure you write it down. Now we will move on to the structure of DNA, chemical and physical structure. So what is the major structure I told you? DNA structure when you want to learn, I told it is double stranded, double helical structure. Okay. Now we will not worry about RNA anymore. RNA will take it almost at the end of DNA. Completion of protein synthesis will take up the RNA. Let us only talk about DNA, Deoxyribonucleic Acid. This I told you double stranded, right? What all it contains? It contains a deoxyribose sugar plus nitrogen base plus phosphate group. One nucleotide. Okay? I told you it is double stranded nucleotide. Double stranded means it will have two strands. First strand I am writing. Okay? And another strand I am writing it here. Here is sugar connected to phosphate. Sugar, phosphate. Sugar, phosphate, and sugar, phosphate continues. Sugar connects the next sugar by adenine to thymine, or cytosine to guanine, guanine to cytosine, or thymine to adenine. So here you have sugar, here you have phosphate. Sugar, phosphate. Sugar, phosphate, sugar and phosphate, and so on. So this looks like a ladder. You can make out that it's looking like a ladder having two strands and the steps in between. These steps are formed by the nitrogen base pairing, and these are the two strands made up of sugar and phosphate backbone. But as you can see, this is the simple structure of DNA, which is showing some just ladder, right? But exactly it is not this way whenever you observe in a detailed fashion. So let me give you the chemical structure of DNA. Let me write a deoxyribose sugar. This is a deoxyribose sugar. Okay, pentose sugar. Here is the nitrogen base. Maybe adenine, guanine, cytosine, anything may be there. Here is a phosphate group. Okay. Next sugar. Okay. Again, there may be a nitrogen base, which is cytosine, guanine, whatever it is. Again, there is a phosphate group. See how I am writing it. This is attached to the next sugar. This is again thymine, whatever it is. It's a phosphate group. Next one. It will continue like this, right? You can observe one clear thing in this, okay? In every sugar, the active carbon is, this is 1, 2, 3, 4, 5 carbon is here, okay? Third carbon is active, and here it is fifth carbon, CH2OH, fifth carbon is active, okay? So, in every sugar, so this is, see, fifth is engaged here, third is engaged here. The first sugar and next sugar are connected by phosphate group, right? And if it is the extreme end of it, here the third carbon is free to react, it is 3' OH end, we call it. 3' because 3' OH group will be there. I have already drawn the diagram and showed you. It will be like this, sugar, okay? Here it will be OH and H, H and H, OH and H, H and CH2OH. This is the fifth carbon, right? This is the third carbon. I said, if it is the starting sugar, this third carbon is free, it is called 3' end or 3' OH end. You can write 3' end also. Suppose this is the other extreme. In between we have millions of such sugars. This is, imagine, the last sugar. In this fifth carbon, CH2OH will be there. No? This is the fifth carbon free. It is called the 5' end. It runs like this, right? Now, another strand connects to this, right? It can't run like this. So another strand runs in a reverse fashion, like this. Cytosine to guanine, they will bind with 3 bonds. Then there will be a phosphate group. Fifth carbon to the third carbon of next sugar. Okay. These two are connected. Again here there is adenine. Again there is a phosphate group. This is again connected to the third carbon. Again here is guanine. Phosphate group next to the third carbon. It is attached to third carbon. This way, here is thymine. You can make out how it is, right? One runs in 3' to 5'. This way, here it is 3', here it is 5'. It runs here in this way. So, both the strands are running parallel to each other because they are forming bonds. Nitrogen and base are forming bonds. But one runs from base to the top, one is top to the bottom, like this they are running. It is called anti-parallel strands. You remember this structure, then only you will understand why it is called 3' and 5' ends. They don't have some other names. 3' refers to the third carbon, free carbon to react. That's a fifth carbon which is free, other end. Same way here is third carbon, is the fifth carbon. So this is the chemical structure of DNA. Right? So remember, this structure will lead to the physical structure later. Okay? First you draw these two. This is the simple diagram. Is called as a ladder like DNA. Ladder like simple form of DNA. This is the chemical form of DNA. Chemical DNA structure. Okay? After this I will give you the physical model of DNA given by Watson and Crick. So draw this. Right? I want you to draw and finish this diagram. Okay, after you are done with this, now we will move on to the DNA structure given by scientists Watson and Crick. For this structure, they were awarded with Nobel Prize. Right? I am going to give you now the structure of double-stranded DNA given by Watson and Crick. We call it as the double helix model. Model of DNA. They explained this through X-ray crystallography. X-ray crystallography method by which they identified the double helical DNA structure. So what you have to do is, first you have to show some two lines. Lightly you have to draw two lines. Then only you have to draw the remaining structure. So one strand first I am going to write. This is the first strand I am writing. Minimum 3 to 4 coils you have to write in this. And this is the thickness of this DNA. Okay, I hope you are getting this. And I want you to draw this and show me. Then many of you are showing it, but still I want you to draw it and show me. This is one of the strand I have given you. Another strand I will give you with another color. Otherwise you don't understand it. This is the another strand. Okay. This is the thickness of the strand. Like this. Okay. Now, two strands are drawn, right? What they contain? They contain on them the sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, and so on. Sugar connects to the next sugar with the help of nitrogen base pairs. Okay? This is sugar, this is sugar, this is sugar, this is sugar. Here is the phosphate group in between forming the strand. Right? This is sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. Okay? So one strand, one part, you have to fill this with all the details. Right? So how do you label? This is the strand. This is the strand. This is the sugar phosphate backbone or sugar phosphate strand. Okay? This is a strand containing sugar and phosphate groups. Okay? Next is another strand also showing the same thing. Okay? This is another strand, sugar phosphate, and here you have adenine to guanine, and adenine to thymine, cytosine to guanine, guanine to cytosine, or thymine to adenine, like this. So everywhere I am showing you the strands like this. So learn the drawing of DNA. This is very important, not for only examination. So beautiful diagram is this to remember and draw this. Okay. Now let's label. What is this portion? It is the nitrogen bases. Nitrogen base pairs there. The pairs of nitrogen bases. Right? Then you show the coiling. One of the strand runs in 3' to 5' direction. Imagine it is 3' to 5'. This way it is running. Okay. Here is the 5' end. One is running 3' to 5' in the reverse direction. Here is the 5'. Okay. First you learn this diagram, then I will give you the details of this diagram. So this structure contains the sugar phosphate backbone, which is the strand. There are two strands. One is running from base to the top, one is running from the top to the bottom. First strand is this, second is this strand, and they have the connectivity between both of them. One sugar of one strand is connected to the next sugar of the next strand by the nitrogen bases. Right? You can see here like this I have drawn. Whenever you are drawing, be careful that you are drawing like this, and you can erase one part wherever it is overlapping. Okay? If you erase it, it looks that as if it is overlapping. Right? Because I am observing students when they are writing, they will mix it so badly, they don't even understand where to erase. Right? So this looks like one overlapping over the other. Right? Okay. Now, you can see here one strand moving upwards, one is moving downwards. Anti-parallel strands. They have sugar, phosphate, nitrogen base. One unit is this. This is one unit. 1, 2, 3. This is one nucleotide. Right? Now, what are the other things to label? Here you can make out each strand. Imagine these are two strands. The two strands are there. They are coiling. They are not straight. Like earlier I told you, It's like a ladder. But later scientists Watson and Crick explained that this is a double helix model given by scientists Watson and Crick. Watson and Crick. They won the Nobel Award for this. They gave the double helical DNA model, right? So they explained that DNA is not just simple ladder-like structure. It is a helically coiled ladder. It looks like a double helix or a circular staircase. Like staircase, if you observe, it will be like this and turning like this. It get turned, right? So first turn, then next turn, like this. Both of the strands will be turning. Now while turning, imagine this is one strand, this is another strand. This is taking one long turn, then the inner one is taking a short turn. Okay? This will get a minor groove, this will get a major groove. Then this comes out. Then this takes a long turn, this takes a short turn. This is the reverse way. Means, imagine from here you are watching. This is the minor groove. One groove is there now. This you can call it as the, this is a major groove. This is a major groove. And its small groove is here. This is the minor groove. Okay. One complete groove is one major groove and one minor groove for both the strands. Mean this strands minor, major grooves here. So from here till here, if you take it, it is a one complete turn of DNA, double strand DNA. We call this as a guide. Guide is nothing but the complete turn of double-stranded DNA. Turns, right? Having one major and one minor groove. Okay? Then, the distance between the nucleotides. See, this is one nucleotide, this is one nucleotide, another nucleotide. One nucleotide, another nucleotide. The distance between these two nucleotides is always 3.4 angstroms. That's the distance between the two nucleotides. Then, a guide consists of around 10 base pairs. Therefore, from here till here, it measures around 34 angstroms. That is the complete length of one guide. That is called as a guide. Guide is involving 10 base pairs. Okay? Then, the width of DNA from here till here, this width of DNA is 20 angstroms. Okay? So that's about the double-strand DNA. It is showing the regular right-handed helix, because the right-handed helix, it turns like this, clockwise direction, right? Apart from this, whatever I have drawn is called as a regular DNA, right? B DNA, right? Sometimes you get Z DNA, which takes a left-handed helix, it turns like this, right? And in this one guide, one complete turn involve 10 base pairs, but there you will get more than that, 12 base pairs you will get it. This is because whatever I have drawn is a B DNA. You have A DNA, B DNA, Z DNA, right? Here, the difference is A DNA, B DNA both are right-handed helices, but Z DNA is a left-handed helix, and the structure is almost the same, but there it undergo the reverse way turning, right? Now, you have to pause this diagram and draw this diagram. This is a very important diagram for 5 months. Okay? So pause the diagram and write the double helix model given by Watson and Crick. They awarded with Nobel Prize. Watson, Crick and Wilkins. Another person is that. His name is Wilkins. These three people share a Nobel Award. Right? What is this explaining? This they gave a structure of DNA through X-ray crystallography. Let me summarize it. What it consists of, according to them, DNA is a double-stranded helical structure. It's not a simple ladder, it is helically coiled ladder-like structure. What all it consists of? The two strands which are there, they are made up of sugar and phosphate backbone. They are the strands, sugar, phosphate backbone, one strand, another strand. Sugar, phosphate backbone, right? And the two strands are connected by the nitrogen bases, in which the pairing will be adenine with thymine or thymine with adenine, with 2 hydrogen bonds. Guanine with cytosine, cytosine with guanine by 3 hydrogen bonds. Okay, that's true. ATGC is base pairing, and they connect sugar to sugar of the opposite strands. Then One strand runs upwards, one strand runs downward, another strand runs downwards, right? Therefore, they are called anti-parallel strands. They are strands running in opposite direction, one from base to top, one top. They are running like this. Okay, anti-parallel strands there. Now, apart from that, each strand will undergo one major groove while turning, one minor groove. If you count for both the strands, then we call it as a gyre. Gyre consists of one major groove and minor groove for each of the strand is called as a gyre. Then each gyre involves around 10 base pairs, right? Therefore, the length of the gyre is 34 angstroms and the distance between the nucleotides is 3.4 angstroms, right? And the width of DNA is 20 angstrom units. Now, let me give you the characteristics of double helix model of DNA given by Watson and Crick, right? So, first characteristic, so all the characteristics I will give you. First characteristic, DNA shows double helix structure, double helix, or it is the right-handed helix, whatever I have written is a BDNA, okay? Second one, the DNA shows, okay, right-handed, many are there, so not only BDNA, many are showing right-handed helix. Second one, the two strands are made up of sugar and phosphate as a backbone. Third point, the two strands run anti-parallel to each other. Anti-parallel is another thing you have to remember. They run in opposite direction, 3' to 5' direction, one from top to bottom, one from bottom to top, right? Fourth point, the double helix DNA shows the nitrogen base pairs in which adenine pairs with thymine by 2 hydrogen bonds and guanine pairs with cytosine by 3 hydrogen bonds, or T2A, C2G. That's a nitrogen base pairing. Next, the DNA will be showing the right-handed helix. It shows a complete turn, we call it as a gyre, which involve one major and one minor groove. Minor groove. Okay. This involves 10 base pairs is equal to 34 angstroms. 6th point, the distance between nucleotides is 3.4 angstroms. Right? These are the basic things to write. Then once you finish this diagram, I would like to give you the remaining characters also. Right? What are the remaining characters? I think I can write it here. 7th point, there is something called the base pair equality rule. Right? There is base pair equality rule. There is a rule. Okay. What is this rule saying? Number of adenine will be equal to number of guanine. Number of adenine will be equal to number of thymine, and number of cytosine will be equal to number of guanine. Means how much ever adenine is there, so much will be thymine. How much ever cytosine is there, so much will be guanine. This is called Chargaff's rule. Chargaff's rule says amount of adenine must be equal to thymine, amount of cytosine must be equal to guanine. That is, A plus T by G plus C or C plus G will be a constant for every organism. Every organism have particular amount of this ATGC content. That is the Chargaff's rule. Actually, RNA will not follow Chargaff's rule because it is not showing equality of base pairs. Right? That's another 7th point. Next, 8th point is these strands, they are having that particular turn. They will show stability in their chemical structure. There are certain chemical bonds present in the DNA which will make the strand or the DNA stable. What are the various chemical bonds present in the DNA? First one is covalent bond. Second one is phosphodiester bond. Diester bond. This is found between sugar and phosphate, right? And third one is hydrogen bond. Hydrogen bond is found between the nitrogen base and nitrogen base. Covalent bond is present between sugar and the nitrogen base, right? This makes the structure of DNA stable, right? So this drawing with at least 6 to 7 points you write, you will get 5 marks. It's a 5-mark question. So what all you are going to write is first the drawing, practice the drawing like this. Then apart from that, writing the points is important and labeling is important, right? Minor group, major group, guide and everything you have to label it, right? So let me list them again. Double stranded DNA. DNA is a double helical structure. It has 2 strands made up of sugar and phosphate group. They run anti-parallel to each other. They show the helically coiled structure like a circular staircase. And the 2 strands are connected, the sugar to the next sugar, by nitrogen bases. Nitrogen bases show base pairing whereby the purine pairs with the pyrimidine. Base pairing here is adenine to thymine or thymine to adenine. And another base pairing is guanine to cytosine or cytosine to guanine. Right? And base pair rule is followed. We call it as Chargaff's rule. According to the rule, base pairs will be having equality. Amount of adenine should be equal to amount of thymine. Amount of guanine should be equal to amount of cytosine. Right? A plus T by G plus C will be constant for every organism. AT, GC content is constant. Then the nitrogen bases pair rule I told you. Guide is a complete turn of a DNA showing one major and one minor turn for each of the strand, right? And the guide consists of totally 10 base pairs, and its length is 34 angstroms because the distance between the successive nucleotides is only 3.4 angstrom units. Then the width of DNA is 20 angstrom units. Whatever I am drawing in the VDNA, there will be ADNA also. Both are showing the right-handed helix. And ZDNA shows the left-handed helix, right? This is the most common one, so I have written this one. Okay, so write down the diagram and some of the points you list and show me later. Okay, then once we have finished with the DNA structure, that is the physical model of DNA given by Wax and Crick, we will have to now move on to how exactly this DNA is present in the chromosome. So in chromosome, I told you it is highly coiled, right? Chromosome is not just simply thread like it is present. It will be coiled. With the help of histone proteins, it get coiled. I hope you have already learned about the nucleosome model. But even you have seen in prokaryotes, if you think. So in prokaryotes, there is no proper nuclear membrane. This is the bacteria. Then nucleus throughout, there is a nucleoid we call it. It is not found spreading throughout. Instead, it will be having one particular manner. It will be arranged. How it is arranged? Why it is arranged like that? Because normally DNA is normally negatively charged thing. It is negatively charged. It will be held together with the help of certain proteins that are positively charged, due to which it will have a particular shape and it will be forming one DNA structure in a one circular DNA fashion. It's not just thrown like this, right? But if you consider the eukaryotes, in eukaryotes, apart from the regular threads, there are histone proteins present. So DNA will be negatively charged, but the histone proteins are positively charged. Histones are positively charged. Normally they contain, most of them are lysine and arginine. These are present in the histone protein. These are the amino acids. These histone proteins are positively charged, due to which negatively charged DNA will wrap around it, forming a nucleosome structure. You know, nucleosome already I have given you. Once we have discussed, there are histone proteins H2A, H2B, H3, H4 present in pairs. They are wrapped by the DNA and are linked by the DNA, on which there will be an H1 protein then again it will go to the binding of next histone octamer. So this we call it as a histone octamer, right? This is the linker DNA. And total nucleosome is a total structure which consists of all this DNA along with the histone octamer. And there are some, this is not the only coiling, right? This is the nucleosome. If at all you think of one chromatin like this, first coils, but it is further coiled like this. I told you multiple coils are there on this. These multiple coils are formed by the binding of non-histone proteins or non-histone chromatin proteins, NHC proteins we call it. These form the multiple coils in the nucleosome model only. We call it as the solenoid model. This is the solenoid model is further coiling of this. This is one coiling has happened. DNA itself has coiled on its own, this double helix model. The DNA is coiled around the nucleosome, that is the histone proteins octamer. This is second coiling. Third coiling is the solenoid model where it is multiply coiled. How it is held together is again by non-histone chromatin proteins are there, they are going to coil around. So due to which there will be two types of chromatins formed. Some are less coiled called euchromatin, some are more coiled called heterochromatin. So this is how DNA is arranged. But all prokaryotes do not contain the histone protein, they contain something else. I told here, some protein which is positively charged, they don't have histone proteins. They have positively charged proteins which will hold the DNA at particular position in the cytoplasm of the bacteria. Right? Now, let's now move on to the next unit. This is how DNA is arranged in the nucleus. Now we are going to talk about the hereditary material. Which is hereditary material in the cell? As I have already discussed and I told you that hereditary material in all the living beings is DNA, I told you. But how they came to this conclusion? Imagine the time when Mendel was there, he did not have any clue that what are the factors, what are the genes, nothing he knew, right? Later things modified. The things were modified after almost 1900. Morgan work and everything was started. But still nobody knew what part in the chromosome or in the nucleus is hereditary. What part is acting like hereditary part of a cell, right? Why we call it, which is that actually the part which is involved in transfer of characters. So what is hereditary factor? So we are now talking about hereditary factor. So actually hereditary factor identification started by one classical experiment, that is on transformation. So transformation experiments, they were conducted by scientist Liederberg. Okay. Scientist Liederberg conducted certain experiments, that is where he identified that there is something is transferring. Suddenly he found like a magic. He was working on one of the bacteria that cause pneumonia, that is Streptococcus pneumoniae. Streptococcus pneumoniae. It is also called pneumococcus bacteria. This bacteria he was working in the lab, right? He was in the lab, he was going through these bacteria. Then he found one sudden transformation happening. One kind of bacteria physically got transferred to some other bacteria. That was very wonderful for him and even for the scientific field. It was a wonderful discovery at that time he found. So what he identified, his experiment, let me give you, right? He was working with two strains of Streptococcus bacteria that cause pneumonia in mouse. They cause pneumonia in mouse, right? Those two strains, when they were plated on the Petri plate, how do you grow bacteria? See, I have to give you all that, otherwise you don't understand what are those. So whenever you want to grow the bacteria, some small Petri dishes you have to take and put the culture media in them and grow them, right? Culture media for the bacteria, they are here put it. One strain was producing the smooth colonies. These are called as the colonies of bacteria. Bacterial colonies, because these are the ones which are forming smooth colonies. Means their surface is smooth. He calls this as the S strain of bacteria. Another one he took. He found that another type of bacteria are forming the rough colonies. These are the rough colonies. And he called, you can take it as capital S strain, and he called this as the R strain. Right? Two strains he had taken for experiment. Now, S strain bacteria form the smooth colonies because these bacteria are capsulated bacteria. They contain capsule around the bacterial covering. It's a covering of the bacteria is made up of one mucopolysaccharide capsule. And when these bacteria were injected into the mice, mice developed pneumonia and it was killed. Mouse was killed because of pneumonia. That means this bacteria is harmful bacteria. Therefore, he called this particular strain of bacteria as the virulent strain. Virulent refers to the bacteria which is harmful bacteria. So, whenever this was injected into the mice, mice got killed because it developed pneumonia. Therefore, he called it as a virulent strain of bacteria. Now, when he took the R strain, R is the rough strain. Rough strains, they contain no smooth outer covering, they have no capsule, their outer surface will be rough in nature. Whenever this was injected in the mice, mice was alive. There was no harm to the mice. Right? That means it is harmless bacteria. He called this bacteria as a non-virulent strain bacteria. There are two strains, one is virulent strain and the other one was a non-virulent strain. So that was the basic experiment. What was the experiment? He started experiment with one kind of bacteria that caused pneumonia in mice. Its name is Streptococcus pneumoniae, or we also call it as pneumococcus also. Both are same. You can write this as, in simple way, you can call it as pneumococcus. Okay. Now, in that two strains he was observing, one strain was forming smooth colonies. Colonies in the sense, the group of bacteria forms small bubble-like things, they are called colonies. If at all you want to think bacteria, they are not so big. In a small petri dish, it will be one small this size. This much is a petri dish, small one. In that you have to put the culture media. Culture media is all the nutrition, whatever required for the bacteria. After that, you streak the bacterial colonies. Take one small bacteria and just inoculate. Put one line or one dot. That's called as a inoculation. Normally they do streaking like this, cross this streak. Then that's how they do the experiment. I am explaining in depth, right? Then one of the bacterial colony was smooth in nature. Such bacteria were having outer covering as a capsule, and because of the capsule, these bacteria will not get affected by the digestive juices or hydrochloric acid of the mouse. Right? Whenever any bacteria, if it enters our digestive tract, and if it enters the tract, if there is hydrochloric acid, most of the bacteria get burnt. But I am talking about those bacteria, those which have outer covering as a capsule. If they enter also, they will be unaffected by hydrochloric acid. Such bacteria will cause disease. Therefore, the smooth strain bacteria was, when it was injected to the mice developed pneumonia and was killed. Therefore, this strain is called, he called it as a virulent strain of bacteria. Then another strain he took, which is the R strain or the rough strain, because it from the rough colonies. When this was injected into the mice did not get pneumonia, it was alive because this is not effective. It was called as non-virulent strain bacteria. Now, this is first set of experiment. Okay. In second set of experiment, what he did was, he took the S strain, but he heat killed them. Right? This bacteria can be inactivated or killed by heating the bacteria. He took S strain and he heat killed this. This is the second. So first set of experiment, you just take it down. This is the first set. I don't want to again mix both the things, right? This was the first experiment he did it. This is how the bacterial characters were. This is the one which is showing how are these bacteria. Okay. Now let me move on to the second set of experiment. How he continued with this experiment, right? The second set of experiment he took in the second group. He took the heat killed S strain. Means S strain bacteria was heat killed, right? Then this, when it was injected with the mice remained alive. No pneumonia. It did not develop pneumonia. It was alive, showing that there is no harm if you heat kill the bacteria and inject. There was no pneumonia found. But next thing, third way what he did was, he took the heat killed S strain, combined it with the live R strain. Two strains he took. He combined them first. One is heat killed S strain, then he combined with the live R strain. Then when he observed mice, when he injected the mice was again killed. So think about it. So he took the second experiment, heat killed S strain bacteria. Means bacteria which is S strain, he made it inactive by heat killing it, and when it was injected into the mouse, what happened? Mice was alive, it did not develop pneumonia. It caused pneumonia when S strain was alive. But now when it was heat killed and injected, there was nothing to mice remain alive. But now, next third experiment what he did was, he took the heat kill S strain, same thing, and combined it with the live R strain, very important thing. He took the live R strain. Then, anyway you know that R strain is not effective, it's harmless. But he found after some time mice got killed. It was surprising to him. So from the killed mouse, when he extracted, he found that from the killed mouse, now whatever killed mouse is there, mice is there, from this he extracted some live S strain bacteria. From where these came? That was the question. Means, when he started the experiment, S strain was harmful, virulent strain. R strain was non-virulent, harmless strain. Now, S strain used to cause pneumonia in mice and it used to kill the mice. But if you take the R strain, when it is injected also, mice remain alive, it was not causing any problems. That was a basic experiment. But this was the experiment, all these experiments are called Griffith's experiment. I'm sorry, earlier I might not have told you, I don't remember. So Griffith experiment we call it. So according to Griffith experiment, S strain is virulent, R strain is non-virulent. Means R strain will not harm, S strain will harm and it will kill the mice. But in the second set of experiment, he took the S strain and he heat killed it, injected in the mice. Then mice was alive. Nothing happened to the mice. That shows that this heat killed S strain is not effective on mice, it is not causing pneumonia. But when this heat killed S strain was combined with the regular R strain, living R strain, and when the two are combined and injected in the mice was killed. This was surprising result for him. And he was wondering why it got killed. He took this mice and extracted the bacteria and observed that there were living S strains. This was the strange result he got. Griffith's result was surprising for himself. He identified that there is something might have transferred from the dead S strain to the live R strain and produced all the new S strain varieties that are going to cause a virulent character. So this he called that what is that something that is transferred. So this indicates that there must be some compound from the dead S strain might have transferred to the living R strain and produced some living S strains. What was this? He did not analyze what is that. He called this as something which is transferred as a transformation principle. We call it as a transformation principle. Means there something that is present in this mixture that has transformed the non-virulent R strain to the virulent S strain, and he called this as a transformation. The process of transformation is defined as the transfer of genetic material from one bacteria to another bacteria through the media, the common media where it is existing there, if they are together. This is called the transformation. So he said there is something which is transferred can be taken as the name as the transformation principle. This has transferred and converted the live R strain into live S strain. From the dead S strain it has transferred. It was called as transformation principle. This is the first basic thing what Griffith explained. This led to the discovery of hereditary material as DNA in the next set of experiment conducted by Avery. Actually, lot of scientists, Avery, MacLeod, McCarthy, Griffith, Liederberg and Totten, they all got the Nobel Award for studying the microbial recombination, bacterial recombination, not bacterial sexual reproduction. They were awarded with Nobel Prize. Actually, it was the whole new things Morgan and all worked in 1900 only. This started at 1926 by Griffith, right? Means they started narrowing down the experiment, right? Like what Morgan explained was the sex linked in the sex chromosome may be having or autosome may be having. All those were the external things. Now they entered into the molecular level, that take out the chromosome out, take the DNA. What does the DNA consist of? It is made up of histone proteins. Okay, take out the histone proteins, take the DNA and work on that. Here they wanted to prove which part, whether it is a protein, is it a DNA or is it RNA which is acting as a hereditary material. Now we know that hereditary material is DNA. But during then, when nothing was the proof for them, they were bringing or narrowing down their experiment. Was very difficult for them, and next experiment was done by Avery. So that I will explain you now. But take it down now, whatever I have explained you. Then the transformation principle was questioned. What is that? Because every cell, bacterial cell may have proteins, it may have RNA, it may have DNA. Anything can be the genetic material, it may be hereditary material. So he left this at this point. Okay. The further experiment was done by other molecular biologists, microbiologists. Their names I will give you now. Second experimental name, that is other people who continued this experiment to identify transformation principle. That is, they identified or molecular characterization they did. Molecular characterization of what? Of transformation principle. Means whatever he said, transformation principle, what exactly it is in molecular base. That was done by Avery. Before Avery, there were two different scientists, MacLeod and McCarthy. These brought some experiment and came to conclusion that maybe proteins are acting as a transformation principle. they had a doubt that maybe proteins are involved or they are working as a transformation principle, or they may be the hereditary materials. But later, scientist Avery, Oswald Avery, his name is Avery. Now I will give you this Avery's experiment. He proved what is exactly his transformation principle with his different type of approach. What he did was, he took separated the proteins RNA and DNA from the S strain bacteria. Okay, then this is one test tube. Okay, this is the second test tube, third one, and the fourth one. 4 test tubes he took. In all of them they kept the R strain live bacteria. These are all R strains. R strain live bacteria were there. Okay. Now to the first one they added the protein of S strain. This is the proteins of S strain. S strain. Second one they added RNA of S strain. So S strain RNA, S strain protein they added. Then the third one they added DNA of S strain. Last one they added DNA of S strain with enzyme DNase of S strain added. See, 4 type of experiments. So first one, this is the first one, second one, third one, fourth one. So what happened in the first one, when the proteins of the S strain, when they separated and added, they found no S strains. There was no S strain. They remained as R strain, which explained that protein is not involved in transfer of characters. To the second one, when the S strain RNA was added, then also there were no S strains. So RNA is also not involved in genetic transfer. But wherever DNA of S strain is added, they found all S strain recombinants. There were new S strain recombinants were there. So here we had S strains. Right? That means DNA is involved in genetic transfer. To really confirm whether DNA is only involved, what he did was, last experiment, DNA along with the DNase enzyme. So what is DNase enzyme? Any enzyme that breaks down the DNA is called DNase enzyme. Right? They took the DNA and DNase enzyme they added, they mixed and they transferred to this. Then also there were no S strains. What do you mean by this? If DNA is alive, only it will produce S strains. Otherwise, no S strains are formed. This means the genetic transfer or the transformation principle what Griffith explained was nothing but the DNA. And even they used protein with the protease enzymes. They break down the proteins and added, or in the same one where they added complete S strain, full S strain means all the proteins, RNA, everything they added. They added protease enzymes, they added RNase enzymes, retained the only the DNA, then it was showing the new recombinants. But when they are alive, the S strains were added with all this, you cannot identify. If at all you add one the complete bacteria, so many bacterial components are there. How do you identify this is only transferring the character? Right? Imagine there are a lot of students sitting. Somebody is making murmuring sound. How to identify that? Right? What you have to do is you have to separate them off. Right? Each one individually separate and you can make out where is that coming from. Same way, one bacteria consists of so many components. Right? It may be a cytoplasm, it may be a protein, it may be a DNA, it may be RNA, it may be anything else in the bacterial components. There are the micromolecules, the molecules, organelles of the cell are there. To identify which is that, every experiment was 100% useful. Because of this only they could conclude that whatever transformation principle was given as a question mark by Griffith, answer was given by scientist Avery as the DNA is the one which is called the transformation principle. That's how it is one of the important investigation discovery happened during almost 1933. This got the 1933 to 44 that this was concluded. So therefore, Avery's experiment clearly proved that whatever they called transformation principle, that means what is transformation principle? Get back to the previous one, previous experiment. What was it that? The transfer of R strains to S strains by adding heat-killed S strain. There is something which might have transferred. He gave it as a transformation principle. And the answer here given as the transformation principle given by Griffith is nothing but DNA. Therefore, DNA is the hereditary material. So this is the proof that hereditary material in every living cell is DNA, neither RNA nor proteins nor any other cell organelles or cell cytoplasm. Therefore, this is a major discovery happened. After this, another experiment was done by Hershey cheese. I will explain that later. Now let me list the whole story. What is that? This is the whole experiment started to explain which part of the cell is hereditary. Because we say that genes are transferred from parents to offspring because we have highlighted it. To be a scientist, if you are a scientist sitting in the lab and working on this, you will get a doubt. If at all somebody says that DNA is hereditary or genes are transferred, how do you see? You are not seeing genes, you are not seeing the DNA. You have to conduct some experiment where DNA is involved and other parts are involved, then you have to prove it. This proving of the experiment was done by initially by Griffith. Griffith was working with some bacteria named Streptococcus pneumoniae. This bacteria caused pneumonia. This bacteria has two strains. How you differentiate strains? Based on formation of colonies in the culture media. One bacterial colony was smooth colony, surface was smooth. Another bacterial colony was rough because it has no smooth, means the finishing was not smooth. Then wherever there is a smooth strain bacteria, he named it as S strain bacteria. And the smoothness of the bacteria is because of the presence of capsule around the bacteria. They are always capsulated bacteria. So such capsulated bacteria are normally harmful, right? So therefore, when S strain bacteria was injected in the mouse developed pneumonia and was killed. Mice killed because of pneumonia by injecting the S strain bacteria. Second, they took R strain. And when R strain was injected into the mouse, mice remained alive. There was no problem to the mice. That means it is ineffective. It is not causing disease. Now, next thing, S strain is now causing pneumonia. It is called as the virulent strain. R strain was harmless. It was called non-virulent strain. Next is second set of experiment. He took the heat-killed S strain. Then he injected into the mice. There was no infection to the mice. Mice remain alive. It means after heat killing, that might have lost its virulent character. But in the next experiment, he took the heat-killed S strain, mixed it with the living R strain, this non-virulent strain. Mixture of that was injected into the mice. But very surprisingly, he found that mice was killed again. And what happened? It was like a magic to him. Then he started analyzing. When he taken out the bacteria from the dead mouse, he found that there were S strains produced. Then how this transfer happened? Although when he took the heat-killed bacteria, it was not working. Heat-killed S strain was not virulent, but in mixture there was formation of S strains. Then he came to a conclusion that there must be something that have transferred from S strain, which is heat-killed, to the living R strain and produce new combinations, which are called the new recombinants, might have formed. That all became, the children of that became all S strain virulent ones. What is transfer? He called that as a transformation principle. Something might have transferred. Later, that experiment was done in 1926. Now, almost in 1933 this started by the three different scientists. Actually, McCart and McLeod. They first did some experiment and they concluded that protein must be involved in the transfer of characters. But clear explanation was given by Avery by disproving they are not proteins by conducting another experiment. You take this experiment, drawing above. He took 4 vials. In the first one he transferred all of them, he took the R strain live. To the first one he added protein with proteins of S strain. Second one he added RNA of S strain. Third one he added DNA of S strain. Next DNA with DNase enzyme he added. Right? Then in the first one there were no recombinants of S strain. So protein is not responsible for genetic transfer. In second one RNA was added. There were no S strains, showing that RNA is also not involved in the genetic transfer. There is no transformation. This is not a transformation principle. Then when DNA was added in this vial, he found some S strains. These S strains indicate the DNA involvement in genetic transfer that might be the hereditary material. But last one will prove 100% that DNA was only involved, where he took DNA along with the DNase enzymes. DNase enzymes are those enzymes that will break down DNA, they kill DNA. When he added it, of the S strains, DNA with the DNase enzyme was added, there were no S strains, meaning that if there is no DNA, there is no genetic transfer, there is no transformation principle happening. So what do you mean by this? It clearly indicates that transformation principle is only DNA, not the RNA nor the proteins. If DNA is not there, there is no transfer of genetic material. This clearly indicates that hereditary material in organisms is DNA. It is a 100% full proof that DNA is a hereditary material. These two experiments you have to remember, write for 5 marks. Either Griffith separately for 2 marks, Avery separately for 2 marks, or together DNA as hereditary material with Griffith and Avery's experiment. You have to write fully for 5 marks. Okay, I hope you have understood, and listen. Understanding of molecular biology is a different way compared to the regular parts of the biological sciences. Therefore, genetics you have to be more careful in understanding, analyzing. You should have a lot of logic and understand, read between lines, right? So that is how you can get mastery in molecular biology. It is mainly experimental genetics. You are observing. I am explaining experimental genetics, not a normal one. They have done it, they have worked on it. Right? So let's meet in the next class with new experiments. Okay, see you then. Bye.